Good morning children. Today we will discuss the topic nucleic acids. Okay. Now where are these nucleic acids found in your body? The nucleic acids are found in your body in the nucleus. Okay. In the nucleus there is chromosomes on which you will find the nucleic acids. The nucleic acids are of two types, that is DNA and RNA. So in the nucleus of your cell, every nucleus of your cell will contain chromosomes, and these chromosomes are made up of number of nucleic acids, and these nucleic acids will be deciding the how the hereditary character should be passed from one generation to another. Okay. So nucleic acids are actually the hereditary material which is present inside the nucleus. ठीक है. Now nucleus are also called as biopolymers. That means number of nucleotides will be joined together to form the nucleic acids. Just as in the proteins you find that number of amino acids are joined to form the protein, or in the carbohydrates you will observe that number of glucose molecules or fructose molecules or together they form the carbohydrates. Similarly, the nucleic acids are actually the biopolymer of the monomer nucleotide. So ये biopolymer है, biopolymer which is called as polynucleotide. Polynucleotides are made up of monomer unit nucleotide. Polymers क्या होता है? जैसे polythene, when we say polythene is a polymer. Actually polythene is the big molecule which is made from smaller molecules like ethene which is CH2. Number of CH2 molecules are added to obtain polythene. Similarly, here also this is a biopolymer where you have the monomer unit of this biopolymer as the nucleotide, और बहुत सारे nucleotides together form the polynucleotide which is called as the nucleic acid. Okay. So now first what is nucleotide then? Nucleotide, usko general representation is this. What is the general representation of a nucleotide? It has three parts or three components are there. One is the base, then you have a sugar, and you have the phosphate ion. Okay. So when you hydrolyze a nucleic acid or a nucleotide, okay, nucleotide, when you hydrolyze it matlab you carry out the hydrolysis with the help of water. What are the substances you will obtain? One, you will obtain the base. Second, you will obtain the pentose sugar. Okay, what is it? Pentose sugar. And you will obtain phosphoric acid. Phosphate ion, that will also do. So these are the three components of the nucleotide. Generally we represent it this way. Base is bonded to sugar, sugar is bonded to phosphoric acid. Okay, ये एक nucleotide का representation है. Number of nucleotides which are joined together by a phosphodiester linkage, we call that as a that we will discuss later on, phosphodiester linkage. Number of nucleotide, you can see this is one particular nucleotide, है ना? This is one particular nucleotide, and this is joined with another nucleotide, you can observe here. So number of nucleotides are joined together to give you the nucleic acid. ये देखो nucleic acid का यहाँ पे भी open है, यहाँ पे भी open है, which indicates that there are molecules on either sides, the molecules of a nucleotide. So this is what the nucleic acid looks like. It has three components. When you hydrolyze it, okay, when you hydrolyze, you will get base, pentose sugar and phosphoric acid. Clear? Okay. Now nucleic acids, when I say, they are two types, DNA and RNA. What is DNA? It is deoxyribose nucleic acid. okay, and RNA is ribose nucleic acid. ठीक है, फर्क क्या है डीऑक्सीराइबोस न्यूक्लिक एसिड एंड राइबोन्यूक्लिक एसिड में? हियर द पेंटोस शुगर, नो, इट वेरीज. Pentose sugar present in DNA. This is the sugar which is present in DNA. The variation is here only, the presence of sugar. This is the pentose sugar which is present in deoxyribose nucleic acid. ये deoxyribose pentose sugar है. While in ribose RNA, this is the sugar which is present in RNA, that is ribonucleic acid. तो यहाँ पे the sugar present is ribose, beta D ribose, here beta 2 deoxyribose. फर्क क्या है इन दोनों में? What do you observe? Is there any change? Here also you have CH2H, here also CH2H, OH and H. In the second carbon, can you see, observe here, in the second carbon, in case of deoxyribose, it is both H. While in case of ribose, it is H at the plane above and plane below it is OH. Rest everything is remaining the same. So this is only the difference between the ribonucleic acid, pentose sugar in ribose nucleic acid and pentose sugar in RNA. okay? ये शुगर में डिफरेंस होता है. और कौन से कार्बन पे डिफरेंस है? कार्बन नंबर 2 पे डिफरेंस है. Followed this much part. okay? What did I explain just now? Nucleic acids, they are biopolymers. They are made up of nucleotides. What is nucleotides? It has 3 components in it: base, pentose sugar and phosphoric acid. okay? This is the representation of a nucleic acid where number of nucleotide molecules are joined together. okay? When I say nucleic acid, 2 types hai. DNA. DNA is the nucleic acid which is existing in our body, while RNA is the nucleic acid which may be existing in a virus or some bacteria. okay? So nucleic acid which is existing in our body, even RNA will be existing in our body. The DNA is the one which is carrying the hereditary material. okay? Variation है इन दोनों में. It is only that the pentose sugar में at the second carbon there is a change. There is an OH group present in the ribose sugar, while there is no OH present at the second carbon in the deoxyribose sugar. Got it? So you can note this down first. The next we will discuss is about the bases in the nucleic acid. 2 different kind of bases are there. One is purines, other is pyrimidines. ठीक है? Purines include 2 bases, that is adenine and guanine. We represent adenine by A and guanine by G. ठीक है? Adenine has this kind of a structure where there is 2 cyclic structures present. One is a hexagonal structure, other it is joined with a pentagonal structure. The number of carbons you can understand. The first carbon is here, second, third, fourth, fifth. This is how the numbering is done. 7, 8, 9 carbons are here. Okay. Similarly, here you have the first carbon is at this position 1, 2, 3, 4, 5, 6, 7, 8 and 9. So therefore, the structure of guanine, adenine will contain 2 cyclic structures bonded together. In pyrimidine, this is another kind of base which is including uracil, thymine and cytosine. It has single cyclic structure. Hexagonal structure is present in all of them. These are the structures. You are not required to learn the structures, you just have the understanding. In DNA, you will find the bases present are adenine, guanine, thymine and cytosine. Okay? While in RNA, you will have adenine, guanine, cytosine, but instead of thymine, you will have uracil. So uracil will only be found in RNA. ठीक है? or thymine will only be found in DNA. ठीक है? And cytosine both RNA and DNA. Okay, it will be found both in RNA and DNA. Similarly, adenine, guanine will also be found both in RNA and DNA. Okay, both in RNA and DNA. Followed everyone? Okay, uracil is represented by U, thymine is represented by T, and cytosine is represented by C. So, these are the bases which are present in the nucleic acid. Whenever we represent bases, it may be either of them. It may be an adenine, it may be a guanine, it may be a cytosine, it may be a thymine. If it is a DNA, if it is a RNA, then it may be uracil. So, these are the 5 bases which you observe here. Okay. Pyrimidines will have hexagonal ring structure, while purines have heterocyclic structure तो दोनों ही है. Heterocyclic meaning what? Where you have other atoms than carbon in the cyclic structure, then it will be called as heterocyclic structure. Here you will find 2 rings together in purines, while here you have a single ring. Okay, ये भी heterocyclic है क्योंकि here you have apart from carbon there is nitrogen, oxygens are there in the ring. So therefore it will be heterocyclic rings. ठीक है. So basic thing you have to understand here is that A, which is represented by adenine is represented by A, guanine represented by G, uracil with U, thymine with T, cytosine with C. In DNA, which are the bases present? The DNA will have bases adenine, guanine, cytosine and thymine. While RNA has adenine, guanine, cytosine and uracil, not thymine. Okay. So, this is what you have to remember for bases in the nucleic acids. Previous we have seen what is a nucleoside. Now, we will see what is a nucleoside. Nucleoside will have only 2 components in it, that is the base and the glycosidic bond. Okay. Base we number the different elements as nitrogen mein number 1 you will be giving, that is always the nitrogen with number 1 will be bonded to the pentose sugar. So, here 1, then 2, 3, 4, 5, 6 you will observe here. Then the carbons of the pentose sugar will be given the numbers as 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. ठीक है. And these two are bonded together by a glycosidic bond. So, this is about nucleoside. When you hydrolyze a nucleoside, you will get only 2 components in it, that is the base and the pentose sugar. ठीक है. Let us observe the formation of a nucleic acid. Whether it is a DNA or an RNA, it does not matter, but this is how the formation of nucleic acid will take place. This is the phosphate ion, this is the pentose sugar and this is the base. The pentose sugar we have not indicated the hydrogens or the OH because it can be an RNA, it can be a DNA. Okay. If it is an RNA, then you will have an OH here and a H here. If you have a DNA, then you will have H and H here. ठीक है. So, these are 2 nucleotides. This is one nucleotide, this is the other nucleotide. They will be bonded together by a phosphodiester linkage at carbon 5 of one of the nucleotide and at carbon 3 of the other nucleotide. Are you observing? देखो यहाँ पे एक phosphate ester linkage will be formed. Okay, this is the 5th. ये जो linkage है, this is formed between which 2 carbon atoms you can observe here. See, this is the 5th 5 dash carbon here. Okay, 5 dash carbon के साथ ये phosphate linkage है ना. देखो ये तुम्हारा फर्स्ट कार्बन है, ये सेकेंड है, ये थर्ड है, ये फोर्थ है और ये फिफ्थ है. सो दिस इज द फिफ्थ कार्बन with which the phosphate ion is there, and that phosphate ion will be bonded to the third carbon of the other nucleotide. Okay. so you will observe a phosphodiester linkage being formed. यहाँ पे you understand this phosphate has been moved here. So this is the phosphate linkage which you are obtaining, which is occurring between the 5' carbon of one of the nucleotide and 3' of the other nucleotide. इसका phosphate linkage again it is here. This again will be linked to the third carbon of another sugar, pentose sugar. ठीक है, so that is how a number of nucleotide together will make the nucleic acid. This is only 2 nucleotides, that is dinucleotide है यहाँ पे. Similarly, you can join one more. You can see, you can use the one more nucleotide where you have the CH2OH group and get bonded with the third carbon. Okay, in your books you can note down that way. ये OH जो है, this is the third carbon, no? 1', 2', third carbon. This will be bonded to another nucleotide of this kind at the 5th position. ठीक है, so that you can show here. So this is how the nucleic acid formation is taking place. Between the 2 nucleotides you have a phosphodiester linkage. This is what we call as the phosphodiester linkage. Okay, ये जो O minus है, it will get bonded to the H, okay, of the third carbon of one of the nucleotide, third carbon of the pentose sugar in the nucleotide, and the 5th carbon in the nucleotide of another, in the 5th carbon of the pentose sugar in the other nucleotide. Okay, so this is how formation of nucleic acid will take place. You can note this down, it's there in your textbook also. Now we will discuss the nucleic acid DNA. Okay, we will discuss the structure of DNA. DNA has a double helical structure. Okay, it has a double helix structure. ठीक है, तो अगर हम उसको बनाएं, so it will be in this manner. ठीक है, so you can observe that there is a double helical structure which are coiled in this manner. ठीक है, so this is how you will find your double helix structure of DNA. It was found out by Watson and Crick that it had a double, DNA had a double helical structure. अब ये जो double helical structure में there are 2 strands you can obtain. So each strand, no, all of these strands, 2 strands you observe. At these strands you will have phosphorus, which is the part of the nucleic acid, and then you have the sugar. Okay, then you have phosphorus. I will write the sugar name as D because in DNA the sugar is deoxyribose. Okay, so P, D, P, D, P, D. Okay, so here also D, then P, then D, then I have P, D, this way. ठीक है, उसी तरह से on the other strand also you will have D, P, D, P, you have D, P, D, P, this way. ठीक है, अब base is joined, base is present in this nucleotide or the nucleic acid in the inward side. And where is the base joined with? It is joined with the sugar. So if this is your sugar which is bonded with the base A, then which is the corresponding base it will be joined with the other strand? It will be thymine. Always remember, the base A will be joined only to thymine, and these 2 bases are joined together by a dotted line which indicates that there is how many hydrogen? There are 2 H bonds. Okay, these 2 H bonds are joining the A and T bases which are present on the opposite strands. ठीक है? If your base is G, then it will always be bonded to C, which is cytosine, and there will be 3 hydrogen bonds present. Okay, it will have 3 H bonds present. So 3 H bonds between G and C, 2 H bonds between A and T, this is fixed. Okay. Now suppose there is a sugar here D, and if it is bonded with C, then again the complementary base will be G which is bonded to the, तो ये हमेशा इसी तरह से बॉन्ड होता है, अगर यहाँ पे C है तो यहाँ G होगा. Okay. Similarly, if here you have sugar with A or with thymine, then always it will be bonded with A, which is the complementary base of T. तो इस तरह से always you will find that one strand it will be arranged in a complementary manner to the other one. कभी भी दोनों जो strands है वो same नहीं होते, they won't be same, they will be complementary to each other. क्योंकि एक strand पे अगर A और G है तो दूसरे strand पे T और C ही होगा. If एक strand पे T and C is there, the other one will have A and G. So therefore, these two strands will be complementary to each other. Other thing to be noted here is that A and T will be always joined with the 2 hydrogen bonds, while G and C will always be bonded with 3 hydrogen bonds. So these are the things you should understand in DNA. DNA में the sugar is deoxyribose, ठीक है. The bases which are present in DNA are 4, that is adenine, thymine, guanine and cytosine. Okay. Adenine will always be bonded to thymine. Adenine is a purine base and thymine is a pyrimidine base. ठीक है. Similarly, guanine is a purine base and cytosine is a pyrimidine base. Okay. So bonding between G and C will always take place by 3 hydrogen bonds, while A and T will always take place with 2 hydrogen bonds, and they will be complementary to each other. इस तरफ A है तो उस तरफ T होगा, इस तरफ T है तो उस तरफ A होगा, that way. So this is the double helical structure of DNA. RNA, the structure is not shown in your books, but RNA will have only single strands. और ऐसा एक पेयरिंग प्रिंसिपल जो कि डीएनए में हम ऑब्जर्व करते हैं, ऐसा कोई पेयरिंग प्रिंसिपल आरएनए में हम ऑब्जर्व नहीं कर सकते, बिकॉज इट इज सिंगल स्ट्रैंडेड. इट विल हैव बेसिस एनी हाउ अरेंज्ड. Okay, so this is RNA structure which we are not learning in detail, that you may learn it when you are going for your higher studies. DNA, this is how the structure is. There are 2 strands, therefore it is called as double helix structure, and each strand will have a complementary bases on them. What do you mean by complementary bases? The A will always be, A and T are complementary bases, C and G are complementary bases. ठीक है, तो इस तरह से ही अरेंजमेंट होता है. So this is regarding DNA structure. All of you should read the textbook thoroughly with DNA. Also nucleotide, nucleoside, ये सारा you may note down the difference between nucleotide and nucleoside. You may note down the difference between DNA and RNA. What are the differences between DNA and RNA? Few of the differences we can just discuss now. DNA, it is having 2 strands, while RNA has only 1 strand. DNA, there is no pairing, DNA will have a pairing principle where adenine will be bonded to thymine and guanine will be bonded to cytosine, while in case of RNA there is no pairing principle since it is a single strand. Okay. The sugar present in DNA, it is 2-deoxyribose, while if the sugar present in RNA, it is the ribose sugar. Okay. The bases which are present in DNA are adenine, thymine, guanine and cytosine, while the bases present in RNA are adenine, guanine, cytosine and uracil. So you have got so many differences. ठीक है. So these are the difference between DNA and RNA. If you hydrolyze DNA, which are the bases you will get and which are the pentose, or what is the product of hydrolysis of DNA? The product of hydrolysis of DNA will be, DNA has a pentose sugar, DNA has bases in it, DNA has the phosphate ion or the phosphoric acid. So when you hydrolyze DNA you will get all these, phosphoric acid भी मिलेगा, पेंटोज शुगर भी मिलेगा जो कि डीऑक्सीराइबोज शुगर है, और इक्वल अमाउंट ऑफ एडेनिन एंड थाइमिन, गुआनिन एंड साइटोसिन, क्योंकि वो इस तरह से ही बॉन्डेड है. इक्विमोलर अमाउंट ऑफ एडेनिन एंड थाइमिन, इक्विमोलर अमाउंट ऑफ गुआनिन एंड साइटोसिन. ठीक है. जबकि आरएनए अगर हम हाइड्रोलाइज करते हैं तो हमें पता नहीं चलेगा. वी विल गेट DNA, this ribose sugar, you will have phosphoric acid and you have the bases, but what is the amount of bases we are not able to find out, because there is no pairing principle present in RNA. Okay. So these are few things which with respect to nucleic acid which we could discuss. Vitamins and hormones, enzymes we have already discussed in the chapter number 5. Vitamins, you can just go through that table. There are different vitamins which are useful to us in our body. Vitamin A, B, C, D, E, K and H. H is not mentioned in your book, it is biotin. Okay. So similarly, all the vitamins, what are its necessities, why it is needed, what is the deficiency, if its deficiency causes which kind of disease, all that is given in the table, you can go through it. Vitamin A, which is responsible for your eyes, the health of your eye. So it is vitamin A which is retinol, उसका नाम है retinol. Vitamin B है जो कि B1 है, B2 है, B6 है, B12 है. Okay, all of them are there. Then you have vitamin C, vitamin D, vitamin E, vitamin K and vitamin H. Vitamin H, it is not given in your book, it is biotin. Okay. So all these names you can go through it and learn it. Okay. And enzymes also you can read, and hormones you can read it because that is not included in your course. So you can go through it, you will be able to understand it. Okay. If some thing is not clear, you can just tell us, we will let you know. Okay. Thank you.